We started our cancer class last day itself, and today we are continuing with the same. In the preliminary class, I gave you a brief introduction about what is cancer, what is a malignant, and what is a benign cancer, and we briefly discussed the different causes of cancer. So, whenever there is a cancer, this is just a recapitulation of what you have done till now. Whenever there is a cancer, it is evident that there is a mutation. And if you remember the Wolgenstein made model of a bladder cancer, where I had said you that it starts with the mutation of the loss of the APC allele, followed by the activation of the RAS proto-oncogene, loss of a DCC on chromosome 18, and then loss of P53. So it is evident that whenever there is initiation of cancer, there is a first driver mutation. Just a second, let me get my point. There is a first driver mutation. So there is a mutation at the beginning, and this begins in a single cell where a category of gene, it can be a proto-oncogene, it can be a tumor suppressor gene, it can be a mutator gene, is either turned on or turned off, as a result of which the control over cell division is lost. So once the control over cell division is lost, this is the first breakthrough, the first strike, the initial strike, which might be a result of a spontaneous change within a cell, or it might be a result of um, or it might be a result of a change induced by a carcinogen, which can be again of physical or chemical nature. Once the first mutation hits the cell and the genes, proto-oncogene or is turned on to oncogene, then comes a second driver mutation. That means one mutation is not enough. When we studied the case of colorectal cancer, if you remember, where we had studied that stepwise a lot of mutations are involved. So there is this second driver mutation that helps the cell to survive in an environment where there is low nutrient, low oxygen, and low growth factors. So the cell, there's another mutation, and once there is a second mutation, we see that the total control on cell division is lost. The cell is also losing its specialization status. And finally, there is invasion. And once the cell has become lost its specialized status, the cell can now move out from its original primary foci. It can enter the blood vessel, travel through the blood vessel, and then can, as you can see, it can move out, enter the blood vessel, travel through the blood vessel, and then can come out into some other part of the body and result in a tumorous growth there. So this is the roughly the three strikes of cancer. The first is the breakthrough, where the first mutation hits the cell becomes aberrant, and then there are some concurrent mutations, as a result of which the cell becomes more and more aberrant. So that is expansion. And finally, invasion. When the cell is defective enough that it has lost total control on cell division, it has lost its specialization, then the cell tends to move away from the site of its origin. Coming to the 6 important characteristics of any tumor cell. So if you look into the 6 important characteristics of a malignant tumor cell, as I said, the cancer is a malignant one. Then there is self-sufficiency in growth signals. That means the cell is no more dependent on growth signals coming from outside. It becomes self-sufficient. It becomes insensitive to anti-growth signals. So even if there are inhibitory signals, even if there are anti-growth signals which are asking it to stop division, it becomes insensitive to these anti-growth signals. Then evading apoptosis. As you have understood by now that every cell has a cellular life and every cell is programmed to die. And we studied the 2 basic pathways in which a cell is programmed to die. But once a cell attains the attribute of certain of being a cancerous cell, or for being a cell who has evaded cell division, the next thing that the cell attains is evading apoptosis. So it evades the cellular death machinery, the inbuilt cellular death machinery. And once a cell is capable of evading the inbuilt cellular death machinery, it somewhat becomes immortal. So immortalization takes place. So it's now not going to die easily. Sustained angiogenesis. I discussed this once before also, that the cell which has become cancerous, they tend to secrete factors which attracts the development of blood vessels towards them. So that is angiogenesis. So they help in formation of new blood vessels, and this blood vessels moves towards this growing cellular mass, bringing in oxygen and nutrients. So sustained angiogenesis. Limitless replicative potential. As cell loss on cell division is the loss on the control of cell division is lost, the cell is no more, the cellular division is no more under control of a tumorous cell. So you can understand that the cell can go again and again, undergoing many rounds of replication. Finally, tissue invasion and apoptosis, and metastasis. So if the cell has become malignant enough, it does not stays localized in a place. Certain cells then tend to move out, enter the blood, and through blood they further move out to infect other parts of the body. So these are the 6 different vital capabilities of a cancer cell. Now coming to certain, let's highlight some properties of the cancer cell. One is the autocrine growth stimulation. Autocrine growth stimulation means you have studied cell signaling. And during your signaling classes, we understood the signaling can be paracrine, signaling can be autocrine, signaling can be endocrine, and signaling can be juxtacrine. Now let's come to autocrine. Autocrine means that the cells which are liberating the growth factors, these cells are also expressing the receptors for growth factors. They're liberating the growth factors as well as they are expressing receptors for the growth factors. And these growth factors can now bind to these receptors and result in autoactivation of that particular cell from which the growth factors were released. So that is autocrine activation. A cancer cell is capable of secreting growth factors. It is also capable of production of the receptors for these growth factors. Now, once these growth factors are secreted, these growth factors are then accepted by the receptors, also expressed by these cancer cells. And thereafter, as you can see, the cancer cells will become self-sustained and it continues cellular division. This is the different kind of signaling that can be mediated by a cancer cell. You look into the blue molecule is a growth factor produced by the cancer cell. The red molecule is a growth factor produced by other cell, and this one is a receptor for the growth factor. So a cancer cell is producing the growth factor and has the receptor for the growth factor also. So the growth factor can bind to the cancer cell and results in its autostimulation. In some cases, the cancer cells can release growth factor which are uh, taken up by the neighboring cells that are surrounding the cancer. These neighboring cells may be the stromal cells or the endothelial cells, and they act accordingly in response to these growth factors. They liberate growth factors in response to the growth factors received from the cancer cells, which are again accepted by the cancer cells. So that is paracrine signaling. Sometimes growth factors, distance hormone producing cell are capable of producing growth factors, which are often accepted by the cancer cells. And sometimes growth factors are also produced by cancer cells, which can affect distant target cells. So these are different ways in which a cancer cell not only controls its own cellular signaling, but it can control the cellular signaling of the neighboring cell. It can control a cellular signaling pathway of a distant cell. Coming to another important property as we continue on the autocrine growth stimulation. This is another example of autocrine growth stimulation. This is the receptor and this is the growth factor. And whenever the receptor binds with the ligand, then only signal transduction occurs. That is a normal thing. When the cancer cell sets itself in an autocrine loop, that is, it is producing the growth factors, it is upregulating the receptors on its surface. This is the autocrine loop. As you can see, the growth factor receptor signal transduction is occurring, transcription of the required genes. Again, growth factor upregulated, receptor for growth factor upregulated and growth factor produced. So this is autocrine feedback loop. And in this autocrine loop, constantly growth factors are being produced, receptors for them are being produced, they are being accepted by the cancer cells, and this cycle goes on. So this is autocrine feedback loop. The right side picture is a picture of mitogenic factors. When we studied cell cycle, we understood what are mitogenic factors. Mitogenic factors are those growth factors which are capable of initiating mitosis, that is cellular division. One of an important mitogenic factor, a lot of mitogenic factors occur via the signaling process occurs via the receptor tyrosine kinases. You have studied receptor tyrosine kinases. And one of the intermediate signaling molecule available in this pathway is a RAS pathway. When we studied the production of cyclin D via growth factors, there also you might have observed this protein, RAS protein. Now I'm highlighting RAS protein because I'll be coming to one aspect of this particular protein. Let's look into another form of constitutive signaling. That is, the signaling are constitutively going on. In some cases, what happens, mutations might occur. And in such mutations, if the receptor is mutated, multiple ramifications of that mutation might also occur. For example, in normal case, we know that the ligand binds with the receptor ligand dimerization occurs. We have autophosphorylation or trans-autophosphorylation, and the signal proceeds. That is the basic signaling mechanism of that is mediated through a receptor. But at times, as you can see, as a result of mutation, we are having truncated receptors. And the truncated receptors are fixed in a phosphorylated state. As a result of mutation, the truncated receptors, as you can see, are present in a phosphorylated state, as a result of which they become independent of their binding to growth factors. Initially, the ligand or the growth factors attached, and then it converted the receptor into this phosphorylated state. But as a result of mutation, these receptors are present in their phosphorylated state. So the need of growth factors do not anymore arise, and there is constant signaling being percolated from these receptors. So this is one form of constitutive signaling. There is another example, as I showed you, RAS. This is the normal RAS protein. In its inactive state, it is GDP bound. In its active state, it is GTP bound. Mind it, when it is GDP bound, it is inactive. But if it is GTP, it is attached to a GTP molecule, it becomes activated. Mutations in RAS protein results in a form where it remains constantly attached with the GTP molecule. As a result of its constant attachment with the GTP molecule, you can see constant signalings are moving on downstream from this RAS protein. So these are examples where mutations of the receptor or the protein results in continuous constitutive signaling for the cell to go on proliferation. Coming to another important property of cancer cell, that is the loss of contact inhibition. If you look into this picture, this is a normal cell and this is a tumor cell. What happens is that when normal cells are placed in culture medium, the normal cells migrate across the surface of the culture dish until they make contact with a neighboring cell. So as you can see, further, at the moment it makes contact with a neighboring cell, its migration is inhibited and the normal cell starts proliferating, and after a monolayer has been formed, only one single layer, you can see the whole petri dish has been filled with a single layer of normal cells, the cell stops dividing and the cells enters a quiescent state, that is the G0 state. But in case of tumor cells, the tumor cell starts dividing, and once they starts dividing, cell proliferates, and multi-dense layer, one layer of cancer cell, then again another layer of cancer cell, then again. So the division continues. It continues until and unless there is nutrient available in the medium, because these cells are grown in culture, they need to have nutrients for their growth. So they continue to divide and grow irrespective of the density, unless and until there is nutrient in the medium. Whereas normal cell stops their division and enters into a quiescent stage when the whole of the petri dish has been covered by the dividing normal cells. They do not form multi-dense layer. They form only a monolayer. And another important thing is that in case of a normal cell, you can see this is a self-renewing cell, daughter cell, and then when these normal cells have finally divided to terminally differentiated cells. In case of tumor pathway, there are cancer stem cells which give rise to daughter cells. And these daughter cells continue division, and in this division, the dividing tumor cells do not show any differentiated state. So that is another property. And now coming to the model for the development of cancer. This model was proposed by Knudsen. This is a cancer, this is a form of tumor. This is retinoblastoma, where the RB gene, you know the importance of RB gene, you know where RB gene is present in the cell cycle, and you're aware of the exact function of RB gene. Now, if there is a mutation in the retinoblastoma gene, it is a recessive mutation. But if both the genes of retinoblastoma are mutated, then it becomes it becomes the cell becomes aberrant, and as a result, that since both the mutations, both the genes have become mutated, the cells not only becomes aberrant, there's the development of this form of cancer, which is known as retinoblastoma, as you can see. So let's move on. Now, based on this particular category of phenomena observed, Knudsen proposed his two-hit hypothesis. In the two-hit hypothesis, he showed that some cancers can be hereditary, while some cancers can be sporadic. So if in the family there is a history of cancer, then there is a tendency of the progenies to inherit that cancer. That means people with a history, people having a family history of breast cancers, of retinoblastomas, usually have a tendency to be susceptible to cancerous condition more than people who do not have a history of cancer in their family. So this concept was given by Knudsen, and he said that retinoblastoma, as I told you, it's a mutation of the RB gene which causes cancer in the retina. Now, the retinoblastoma can be of two types. It can be hereditary, it can be sporadic. Look at this particular picture. When we are talking about a hereditary retinoblastoma, we have to assume that the individual in which this mutated gene has come, that family has this tendency of having cancers of the retina. So this individual has inherited an allele of retinoblastoma from either of the parent, which is mutated, and has inherited a normal allele of RB from either of the parent. So as you can see, there is one wild type allele or normal allele of the RB gene and one mutant allele of the RB gene. If there is any sudden mutation occurring further, if there is further any sudden mutation occurring, just a second. And if this particular gene is mutated, As you can see, homozygous mutation of retinoblastoma resulting in tumors in the retina. So when there is this hereditary retinoblastoma, cases of hereditary retinoblastoma present in a family, individuals are more susceptible to this cancer because only one mutation is capable of converting a normal cell to an aberrant cell. But in case of sporadic retinoblastoma, where the family do not have a history of retinoblastoma, an individual has two normal alleles of retinoblastoma. There is a first mutation, and the first mutation results in the loss of the functional RB allele and the RB is mutated. But as I said, since RB is recessive, this is a normal phenotype. Now, for this individual to have retinoblastoma, a second mutation is required. So there is a loss of and the second mutation results in the loss of function of the second normal RB allele. And once there's a second mutation in the wild type RB allele, then only the person develops this retinoblastoma. So from this concept, Knudsen proposed that in cases of hereditary cancers, as exemplified by retinoblastoma, one mutation is enough to change a normal cell to an aberrant one, because a normal cell already contains a mutated RB allele. But in case of sporadic cancers, as exemplified by sporadic retinoblastoma, two mutations are simultaneously required to change the two normal wild type RB allele into two defective mutant RB allele. So this is sporadic retinoblastoma. Depending on the family history, those who have a case of cancer in their family are susceptible to cancers more than those individuals who do not have a history of cancers in their family. However, one thing to be kept in mind that all cancers are not inherited. Some cancers are hereditary, but all cancers are not hereditary. For example, retinoblastoma can exist in a hereditary retinoblastoma form and a sporadic retinoblastoma form. So with breast cancer, but all cancers are not hereditary. Now coming to another important aspect of cancer. So we have done the properties of cancer. We did the causes of cancer. We understood malignant and benign. We did metastasis and invasion. And finally coming to the next part, that is genes and cancer. You have all studied central dogma. And to reiterate, a gene is nothing but a segment of DNA that is capable of giving a biological product. And in these cases, the biological product is usually a protein, because we are talking about genes which are now controlling functions. So the biological products are mainly proteins. Based on the number of genes associated, based on the types of genes associated with cancer, they can be broadly classified into three broad types. One is the proto-oncogene. Proto-oncogenes are those genes, as you can see, which are associated with cell survival or proliferation. That means somehow they are associated with cell cycle pathway. Okay. Or they are associated with cell signaling pathway. For example, we have already seen one proto-oncogene, that is RAS is a proto-oncogene. It is an intermediate signal transducer in a lot of signaling pathways, and it acts as a proto-oncogene. Because once it is mutated, it gets converted to oncogene. Next is coming the tumor suppressor gene. You have studied examples of tumor suppressor gene. The two famous examples of tumor suppressor gene you have already studied is P53 and retinoblastoma. Both these genes are associated with cell cycle, and both of them, if you remember their function, inhibit cell proliferation if the cell has become defective. So they control cell proliferation. In case of any defective arising, they prevent cellular proliferation. In fact, P53TSG, tumor suppressor gene P53, not only prevents cellular proliferation, it checks the genome, and depending on the damage in the genome, it can instruct the cell to promote apoptosis. So tumor suppressor genes are apoptotic proteins, promoting proteins, inhibitors of cell cycle progression. They act in the checkpoint pathway proteins that assess DNA and chromosomal damage. And to some extent, they are the components of the signaling pathway that restrain cell proliferation. The last category is the genome maintenance gene. The genome maintenance gene are those genes which are involved in, as you can see, repair or prevent DNA damage. So these are the DNA repair enzymes. Now, when we are coming to proto-oncogenes, let's move to proto-oncogenes once more. These are anti-apoptotic proteins, components of the signaling and the signal transduction pathways that results in proliferation. Now, in case of proto-oncogenes, this is important. Gain of function mutations allow unregulated cell proliferation and survival. Proto-oncogenes are usually dominant in nature. That means we just now studied the example of retinoblastoma. What you saw, if there is single mutation in the retinoblastoma allele while the other allele is normal, we are seeing a normal phenotype. That means it is a recessive gene. But in case of proto-oncogenes, a single mutation is capable of making the cell an aberrant one. So these mutations are genetically determined and, these mutations are genetically dominant as compared to the tumor suppressor genes where the mutations are genetically recessive. So this is one big difference between the proto-oncogenes and the tumor suppressor genes. If I compare them with the genome maintenance gene, here also the mutations are genetically recessive. Now coming to this point, gain of function and loss of function, I'll explain it in the next slide. But before moving into the next slide, I just want to touch this point, that origin of mutations. When we are talking about proto-oncogenes, the mutations may arise from point mutation, chromosomal translocation, chromosomal amplification. We'll see all these properties. And when we are talking of tumor suppressor genes, they again may arise from deletion, point mutation, or methylation. Now let's move to the gain of function and the loss of function. What do we mean by gain of function? This is a normal cell. I'm talking about a proto-oncogene. There's a single mutational event. And the single mutational event causes a mutation in one allele of the proto-oncogene. That one allele of the proto-oncogene mutated, when mutated, is capable of becoming aberrant enough to result in the conversion of a normal cell to abnormal cell. So a single mutation has converted the proto-oncogene to oncogene. And since the oncogene has been activated, it promotes cell transformation. That means the activity of that protein has increased. A single dose of mutation has resulted in increase in activity of that particular gene. Imagine RAS. A single mutation, let me just go back to this picture once more. Imagine RAS protein here. RAS is a proto-oncogene. A single mutation in RAS in one of the allele is changing the conformation of the RAS so that it is now attached to GTP constantly, as a result of which signal is constantly going downstream. That means its activity has increased. As a result of this mutation, its activity has increased, and hence the gene has gained function. Its normal function was that it can remain in an inactive state and if required can become activated. As a result of single mutation, it is now constantly in an activated stage. So its functional efficacy has increased. It is now producing constant signaling downstream. So this is gain of function. When we are talking about tumor suppressor genes, I said there is loss of function. That means this is a normal cell. A first mutation event. As I said, tumor suppressor genes are recessive. When there is only one mutation in the allele of the two, there, as you can see, no effect. But if both the alleles are mutated, that means a tumor suppressor gene has lost its function. It is not being able to do what it was supposed to do. Since it has lost its function, the resultant phenotype is a cancer cell. Just imagine retinoblastoma as you have done this. If the function of retinoblastoma is lost, then E2F will not remain attached to retinoblastoma. E2F is free, and a free E2F will go and constantly transcribe the genes required for the entry into S phase. So E2F indirectly is becoming free of the growth factors because both the alleles of RB has been mutated and RB has lost its function. So that is loss of function. Whenever it is a proto-oncogene mutation, it is the overactivity of that particular gene, and the proto-oncogene is thus converted to oncogene. All genes are not proto-oncogenes, remember that. Proto-oncogenes are genes that are involved with the signal transduction pathways and cell cycle pathways. And when we are talking about tumor suppressor genes, we need to have the loss of both the alleles of a tumor suppressor gene to result in an aberrant phenotype, but the tumor suppressor gene is losing its function. So that is about the proto-oncogenes and the tumor suppressor genes. So as I said, so once we have understood that oncogene and tumor suppressor gene, to reiterate once more, proto-oncogenes are genes whose product stimulates cell proliferation. Remember, all genes of the body can never be proto-oncogenes. Only those genes whose product normally stimulates cell proliferation. Tumor suppressor genes are those genes whose product normally inhibit cellular proliferation. And mutator genes are those genes whose product ensure accurate replication and maintenance of the genes. So these were the three categories of the genes we discussed. And now these are some beautiful examples of oncogenes and tumor suppressor genes. We discussed RAS. I showed you what happens when RAS is mutated. So that is that is one of a stable example of a proto-oncogene, and it affects the receptor tyrosine signaling pathway. And in case of tumor suppressor gene, we discussed the colorectal cancer where you had seen that the loss of this tumor suppressor gene initiates the formation of class 1 adenoma, if you remember, and you have studied P53. There are other examples, but these are some known examples which you have already studied. Now coming to the 7 category of proteins that results in cancerous phenotype. If you look into if you look into 1, 2, 3, and 4, mutations in these categories: signaling molecule, signaling receptor, signaling molecule, intracellular transducers, and transcription factors. When mutations occur in this category of molecules, they promote cell growth. These mutations are generally dominant and they give rise to oncogenes. So signaling molecules, signaling receptor, intracellular transducers, and transcription factors. The genes for these four category of biomolecules are basically proto-oncogenes. If mutated, give rise to oncogenes. They are dominant in nature. A single allele mutation results in an aberrant phenotype and they gain function. Their activity increases. Now, let us look into the other categories. So we have here DNA repair proteins, cell cycle control proteins, and apoptotic proteins. Now, I'll come to this protein later on. Let's look into this two category. That is a cell cycle control protein, which function to restrain cell proliferation, and the DNA repair proteins. These two categories are encoded by the tumor suppressor genes. Because this tumor suppressor genes is having a tight control on cellular proliferation and DNA repair machinery. Finally, coming to the last category, the apoptotic proteins. The apoptotic proteins can be again divided into a proto-oncogene category, where a mutation might result in further activation of or the gain of function of the cell, or the apoptotic proteins can also be present as the tumor suppressor genes. So apoptotic proteins fall into both categories. Apoptotic proteins includes both the tumor suppressors that control cell division and promote apoptosis, and also contains oncoproteins or oncogenes that promote cell survival. So these are roughly the 7 categories of proteins that play a vital role in transforming a normal cell to abnormal cell. During the development of cancer, mutation might occur in either any one or it can occur in more than one, because cancer is a multi-process step, and usually cancer is not restricted to the mutation of a single gene. Once cancer occurs in a cell, as we have studied earlier also, consequently a number of mutations are incorporated so that the cell is totally transformed into a malignant type. Now coming to the conversion of oncogenes to proto-oncogenes. Just a recapitulation, while I was talking about this particular slide, I told you that mutations might arise from point mutation, by chromosomal translocation, by amplification. So whenever a proto-oncogene is converted to oncogene, these are the different categories of origin of mutations. So moving to that part, so if there is a proto-oncogene, a point mutation might result into the production of a hyperactive protein. A proto-oncogene, there might be gene amplification, that is doubling of the gene, as a result of which excess normal protein is being produced. You see gain of function in each case. Chromosomal translocation. A segment of one chromosome breaks off and joins another chromosome. Excess normal protein or abnormal hyperactive protein. Local DNA arrangements may also convert a proto-oncogene into oncogene. Local DNA arrangements might include insertion, deletion, or transposition. In each case, abnormal hyperactive protein. And there is insertional mutagenesis. Virals might promote the conversion of an oncogene into a conversion of a proto-oncogene into an oncogene. And again, as you can see, excess normal protein. So whenever there is a conversion of a proto-oncogene to oncogene, what you saw from all these images is that there is always a gain of function, excess abnormal or normal protein, as a result of which the cell is becoming a malignant one. In case of human cancers, the three categories of categories of mutations that has been observed are point mutation, chromosomal translocation, and gene amplification. I'll go over them one by one. The first is point mutation. Let's concentrate on RAS proto-oncogene. As you can see, this is the oncogene and this is the normal. In the case of the normal gene, the product, see the 12th amino acid. The 12th amino acid is GGG, a single nucleotide change which alters the codon 12 to GTC. This single point mutation changes glycine to valine. As a result of this single nucleotide change occurring in codon 12, which changes GGC to GTC, as a result of which the change in amino acid is glycine to valine, results in the production of an aberrant RAS oncoprotein. And I showed you earlier, and this mutated RAS oncoprotein remains always in an activated form. Now, RAS is a very important protein that plays a very vital signal transducer role in the cell signaling pathway, in the cell division pathway. Because RAS is a protein which also controls the production of cyclin D. So if RAS is mutated, that means it is constantly activated, which indicates that there will also be constant production of the growth factors. So that is a point mutation in RAS that results in GGC transition to GTC transition, as a result of which a normal cell becomes malignant, and this point mutation is observed in case of bladder carcinoma. So bladder carcinoma, the causative factor is the RAS mutation and the RAS oncoprotein being converted to the RAS proto-oncogene being converted to the RAS oncogene. Coming to gene amplification, MYC is a gene. It's a gene that is present in the signal transduction pathways. In normal condition, it's producing a product. At times, duplication of this MYC gene is occurring. So the MYC gene is duplicated. As compared to the normal product, you see over excessive normal product. The amount of normal product was low, but in the mutant, we are seeing excessive normal product. So this results in a form of cancer that is known as neuroblastoma. So when we have this MYC amplification, it results in a case of cancer that is known as neuroblastoma. In case of multiple copies of RAS in mouse, adrenocortical tumors are also seen. That is RAS. This is MYC amplification. MYC amplification results in neuroblastoma, but when there is RAS amplification, in case of rats, it has been seen, it develops in adrenocortical tumors. So the gene is amplified, excess production of the normal product. And finally coming to chromosomal translocation. This is chromosome 9, this is chromosome 22. You see, there occurs a breakpoint. As a result of breakpoint, in case of chromosome 922, as a result of the breakpoint, the ABL oncogene is translocated from chromosome 9 to chromosome 22, forming the Philadelphia chromosome. This chromosome is known as Philadelphia chromosome, which results in chronic myeloid leukemia. So this is a fusion. You see the two genes have fused, resulting in the production of a fusion protein. This will result in the formation of chronic myeloid leukemia. Now this gene is known as, this particular chromosome where the 922 translocation has occurred, this is known as a Philadelphia chromosome. Another reciprocal, remember this is a reciprocal translocation. Why reciprocal? Because during translocation, a part of the chromosome of 22 is being attached to chromosome 9. A part of chromosome 22 is moving to chromosome 9, and part of chromosome 9 is moving to chromosome 22. The aberrant phenotype is a result of the production of this aberrant protein. This chromosome is known as Philadelphia chromosome, not the other one. Remember that. Now coming to another translocation, this is also a famous translocation, a reciprocal translocation of 814. You see the c-MYC gene, and this is the immunoglobulin heavy chain gene. So there is a reciprocal translocation. This is found in Burkitt cell lymphoma. Here the chromosome 14q is containing the IgH genes. This is the normal chromosome 14, and the c-MYC has moved on to this portion, and it is carrying the immunoglobulin heavy chain gene is now sitting adjacent to the c-MYC gene. So the c-MYC gene is actually translocated from chromosome 8, you see the breakpoint, and it moves to chromosome 14. This is also reciprocal translocation, but that the reciprocal image is not shown. Let me see that image. Yeah, this is the reciprocal translocation. So you can see 14, 8, c-MYC moves to 14. This is 9, 22. This results in bucket cell lymphoma. 9, 22, 22. Some part of 22 moves here, some part of 9 moves here. We have this BCR ABL fusion protein. So this is Philadelphia chromosome 922 translocation. And this is resulting in chronic myeloid leukemia. And this is 814 translocation. Both are reciprocal, which results in bucket cell lymphoma. And finally, coming to the last part of her class, the tumor suppressor genes. Since we have in detail discussed both P53 and RB, so you are aware of the functional status of both retinoblastoma and P53. As you can see, in case of a tumor suppressor gene, they have compared and showed in this particular picture, tumor suppressor gene, normal growth, one gene mutated, still normal growth, both genes mutated, then only loss of function. In case of proto-oncogene, normal growth, one mutation resulting in hyperactivation of that particular gene, resulting in production of excess product. So that's a comparison between TSG and proto-oncogene. And you are aware of the retinoblastoma. The human RB tumor suppressor gene has been mapped in the chromosome 13. It is a 80 kilobase of DNA that encodes a 4.7 kilobase mRNA that produces a 928 amino acid nuclear phosphoprotein form, that is phosphorylated RB. I'm not getting into the details of how retinoblastoma functions because we have done this in details in our cell cycle class, and you are aware that RB normally keeps E2F sequestered. The moment RB is phosphorylated by cyclin D, E2F is free, and the E2F can then results in synthesis of all those factors needed for the cell to move into S phase. So if the function of RB is lost, you can very well understand that E2F is constantly free, and a free E2F will constantly activate all those genes essential for are required for the cell to move into S phase, and thus E2F is becoming free of the control of cyclin D or the growth factors, because cyclin D is actually controlling RB, which is controlling E2F. If RB function is lost, the control over E2F is also lost. Coming to the next tumor suppressor gene, that is P53. We have discussed in details about P53 also. The P53 is located in chromosome 17. It is a 393 amino acid long amino acid. And this is how the P53 looks. If you see the genetic rearrangement of P53. And in case of P53 also, depending on the presence or the absence of the DNA damage, the P53 can make depending on the presence and the absence of the severity of the DNA damage. If the DNA damage is severe enough, P53 opts for apoptosis. If the DNA damage is not severe enough, then P53 opts for repair, and thus, depending on what kind of DNA damage, the P53 can induce a cell to go for apoptosis or can induce a cell to go for arrest. And we had earlier studied how P53 is controlled by the MDM2 protein. So that is something we have already done. So I'm not getting into the details of how P53 is controlled. So this is about, and finally coming to BRCA1. This is just for your information. BRCA1 is a gene that is associated with breast cancer. There are two genes associated with breast cancer, BRCA1 and BRCA2. This is also a tumor suppressor gene. BRCA1 is located in chromosome 17, producing a 190 kilodalton protein, and BRCA2 is located in chromosome 13. As you can see, BRCA1 has multiple functions. Most of the proteins you have not heard, but there are some proteins you have studied while we discuss cell cycle. It can exert a control on P53. It can exert a control on RB1. You have heard about CHK1. It can exert a control on CHK1, that is checkpoint regulation of mitosis. It can exert a control on HDACs. You have heard about this also because you studied chromatin remodeling in SEM one, and you had heard about histone enzymes, histone deacetylases and acetylases that maintains the chromosome structure. So these are the sum of this. Apart from that, as you can see, there are other genes also BRCA is controlling. So BRCA is basically controlling, present at the genetic crossroad in handling DNA damage. When you inherit a BRCA1 mutation, it increases susceptibility to cancers of the breast, ovary, cervix, uterine tube, uterus, peritoneum, pancreas, and prostate gland. So that is about BRCA1. And finally, coming to mutator genes, as I said while discussing proto-oncogenes, tumor suppressor genes, and oncogenes, to repeat, mutator genes are genes whose products ensure accurate replication and maintenance of the genome. So, an example of a mutator gene is the hereditary formation of hereditary non-polyposis colon cancer. In humans, there is this mutation of 4 genes occurring, which gives rise to HNPCC. Now, these genes are basically mutator genes. So, that is, they help in controlling accurate replication, excuse me, and maintenance of the genome. And the mutator genes, as I said, they are basically the caretaker genes. So if they help, as you can see, the number of functions. So any defect in these caretaker genes, defective DNA polymerase, defective recombination, double-stranded DNA repair is hampered, amplification of gene occurs, aberrant chromosome segregation. All of this will result in chromosomal instability. So this was about the mutator genes. And now coming to some of the famous diseases. Hereditary non-polyposis cancer as a result of DNA mismatch repair. Xenoderma pigmentation as a result of nucleotide excision repair. That means the DNA repair genes are being hampered. Then Bloom syndrome. This is repair of the double-stranded breaks by homologous recombination. So in all these diseases that have discussed here, that has been mentioned here, the DNA repair machinery, the genes for the DNA repair machinery are aberrant. The proteins of the DNA repair machinery are defective. That means the mutator genes are not working properly. Hereditary breast cancer is a result of, it can be a result of mutator genes, or it can be a result of the tumor suppressor gene BRCA1 and BRCA2. And in all cases, most cases, we get variety of cancers. Whenever there is a aberration in the DNA repair machinery, it invariably or usually leads to cancer. So it can be skin carcinomas, leukemias, lymphomas, breast and ovarian cancer. So with this, we come to the end of our cancer class.